Okay, recording. So I want to train about Sanger data interpretation here. So if you're watching this video online and you're not in the room right now, the context would be perhaps you have had a plasmid that you just built and you sequence the plasmid, or perhaps you're doing some experiment and you need to do a Sanger sequencing reaction. So if you need to know about Sanger sequencing, go back, watch those other videos. I'm assuming you know what Sanger sequencing is, the gist of it. Here's what happens when you get the data back. That's what we're going to talk about today. So typically you get back two different types of Sanger data. One is the chromatograms, okay, and the other is just a .txt file with the sequence. Most of the time I'm not looking at the chromatograms. It's like an extra detail. Usually I just start with the text file, figure out what's going on with the text file, and then if there's an irregularity, I can go into the chromatogram and investigate a specific base in the chromatogram. So the chromatograms are useful for resolving ambiguity questions about certain types of data. Is the data good? Is the data bad? What about the data quality at this particular base, etc.? That's when I go into the chromatograms. But for sake of thoroughness, I want to show how one would look at one of these chromatograms. So there is online free software that you can download called Chromas, CHROMAS. You can just download. I literally just downloaded the free viewer on this computer 5 seconds ago from this website, installed it. And so when you get chromatogram data, you can open it up in this program called Chromas. So let me open up some data, just show what it looks like here. Would be okay. So the file type is going to be chromatogram file. So for each, like I said, for each of these sequencing reactions that somebody has done, they're going to get two files, the text file and the chromatogram file. You can open only the chromatogram files with Chromas. Okay. So this is what Sanger data will look like. So you can see these different peaks and different colors. Each color is a color-coded base. So if you see a black peak, that's a G. And if you see a green peak, that's an A. If you see a green or a blue peak, that's a C. And then a red peak is a T. So essentially, again, I'm not going to explain how Sanger works in this video, but essentially strings of DNA come off of a column, and there's a laser shooting at it, and it can read essentially like the last base. And based on the fluorescence at that last base, it can assemble through algorithms this string of bases. Okay, and each of the peak intensity. So if you're looking at the y-axis, which would be peak intensity, and x-axis is the base pair. The height of the peak is a good readout of like quality. Okay, so if you see stuff like this, that's typically going to be bad Sanger data. Sometimes it's still interpretable. Like you will get a text file from data that looks like this, and they will assemble some text sequence. And sometimes that text is relevant and it's good, actually can be good data. But it's bad in a sense that there's a lot of error rate when the peak intensity is very low. So what you want to see in a chromatogram is you want to see a couple different things. You want to see a high peak intensity. You want to see a distinct peaks. So like as quanta, here's peak 1, here's peak 2, and then you would know that whatever, this one was an A, this one was a G. Does that make sense? And what you don't want to see, or maybe you do want to see it in a certain experimental context, but what you don't want to see is a bunch of data that has patterns like this, where you're getting overlapping peaks. That's bad. So there's two types of bad data: low peak intensity, and then like overlapping peaks. And if you look at any chromatogram, you'll see in the very beginning of the chromatogram, the data of all Sanger sequencing reactions is always bad at the very, very start. Okay, so when you're interpreting Sanger data, it's important to remember that if you get a string from 0 to say 500 base pairs, it's important to remember that usually the first 50 base pairs is trash. And typically, like, you know, you're gonna, as you go through this and look at the chromatogram, the peak intensity slowly decreases. And there's a point at which the peak intensity is so low that now the data is unreadable or not properly interpretable. So always at the beginning, there's trash, and always at the end, there's trash in Sanger data. So the key to analyzing Sanger data is figuring out what region of your data represents the good data, and then only interpreting in that region of good quality data. Does that make sense? So if you want to sequence, this is for future reference, or perhaps if you were working with a plasmid, if this was some kind of a gene that you were working with and you wanted to sequence, and it was very, very long, imagine it was 0 to 2000 base pairs, you're never going to be able to Sanger sequence this in one reaction because of this property. You're only going to get about, give or take, 500 base pairs. Actually, I shouldn't say that off the top of my head. I don't know. I actually visualize it by like how many lines of text. Like it's usually about like 10 lines of text in like a paragraph format that you get. That's good data. I don't actually know how many base pairs there are, but essentially, like, it's important to know that if you were going to sequence a long gene, you would have to string Sanger reactions using different primers together such that they would overlap so that you could get the sequence of this whole insert. So you would have to do a Sanger sequencing reaction with a primer here, and a different Sanger sequencing reaction, same template, but different primer here, and then a different primer here, different primer here, different primer here. Okay. And oftentimes what is also very common, if you are actually using Sanger data for some type of experimental interpretation or some important result, it also is helpful to do forward reads and reverse reads. So this same sequence could also be sequenced in the reverse by building primers that bind here in the reverse orientation. So Sanger can go either way because DNA is double-stranded, and this should match this in reverse complement. So you can get Sanger data from either a forward read or a reverse read, and sometimes you'll combine them both to sort of build the rigor into your data. Okay, so more looking at the chromatogram. So this is useful for, like I said, if you're wondering about a certain base, you can search for patterns. So imagine you were worried about a particular sequence of base pairs and you wanted to check out an individual base pair. You can just hit Control Find, and imagine you wanted to find AAC, and you were worried about that last T. You can find it, and it will pull it up. So it just pulled up AAC, GT. So it found that string in my sequence. So this is very useful for, I want to check a particular base pair, and then you can search it. So these data, let me see if I can zoom out here. Yeah, maybe. I can't zoom out. Okay, so we should see as we scan further and further and further into the, okay, so here, this one just drops off. So so what you're seeing here in this scenario where you're trying to get a very, very long read, you're going to see the scenario where your peaks gradually get smaller and smaller and smaller. You're actually seeing something different here, and this is actually because of the context of your experiment. So what we're sequencing here is a very, very small PCR product. The amplicon, what is it, like 500 base pairs? Something like 470. Okay, so that proves that, yeah, you can at least get like 470 base pair reads. So what we're saying you're sequencing is an amplicon. If you're sequencing a plasmid, this is circular. And so this read can just keep going and keep going, although the quality is going to get lower and lower and lower. If you're sequencing from a PCR amplicon, what you're sequencing based off, you use a forward primer. So what you were sequencing was this, and as soon as it hits the end, there's nothing more to write. So it's just going to abruptly stop. And so what you would want to see is you would want to see high peak intensity for the duration of your whole product, which is exactly what you see. And then here you see the cliff, the cutoff, and it's right at about 490 base pairs. So this is essentially proof that you are sequencing the product that you wanted to sequence. Now, the final thing I would point out is if you're ever in a situation where you're checking for a polymorphism, you might find this like in an allele. So if there's a gene for blue eyes, there's going to be a gene that maybe codes for blue eyes but has a mutation, and maybe that codes actually for like brown eyes. I'm just using this as an example. I don't actually know. But if you wanted to see this single nucleotide polymorphism difference, the SNP, say up here it's an A and here down here, and this allele it's a G, what you would see is you'd see the same chromatogram, and then at this single position, that spot would be different. And so if both of these alleles were present in your template, what you would see is a chromatogram that looked like this, where in one spot, it wouldn't be able to tell whether there's an A or a G there. And that's because there's actually both present in the sample. Does that make sense? So if you're looking for polymorphism, chromatograms can be very informative because you can find these regions where you can find a single nucleotide polymorphism and you can actually visually see it in the chromatogram. Okay, but this is typically, okay, so I'll just reiterate, this is typically not what I'm first opening. I'm typically actually like not even looking at the chromatogram unless there is a reason to start questioning or investigating the data. Typically what I'm doing, okay, so let me close this. Typically what I'm doing is I'm straight up opening the text file. So here's seq file. That should be the .txt. Let's see what opens up. Notepad. Yeah, okay. I'm actually going to pull this into Sublime. I've talked about this before. A lot of times there's these annoying like spaces. I will remove those with the Control H. I'll find the new space return carriage key, and then I'll just replace it with nothing. I will copy this, and I will actually analyze. And this is what I'm going to encourage you to do. I analyze all my Sanger data in Word. There are programs to analyze your Sanger data. Some people are going to think I'm old-fashioned. There are like complex programs. The reason I like to do it in Word and the reason I encourage students to do it in Word is because it actually contributes to learning how to read and write DNA. That's actually like an important skill, learning how to interpret and look at DNA sequences and find patterns in the DNA sequences. And many of the algorithms or the computer programs, they might assist you, but they won't assist you in teaching you the foundations that you need to know, if that makes sense. So there's a huge benefit to starting very, very simple and learning how to analyze Sanger data in Word. Plus, if you have a particular important experiment or a particular clone, oftentimes you want to organize all your Sanger data into one file. So for this particular case, this sample was 1A, it was 1A, JFB 1058 A11. So typically, when you're constructing these files, which you should do, you should have a very ordered system of storing and keeping data, okay? And I will always write at the top of some of like a Sanger document, like what I'm doing. So I'm not gonna write what we're doing here because I want to keep it secret. But I would put something like 5.26.2021 the experiment description, I would like describe the experiment that we're doing. I would put the date that I labeled the experiment in my notebook so that this file is associated with that specific date in my notebook. So if I ever am looking through my notebook and I say, oh, I want to find the Singer data for this, I know how to find it. And I would probably give it some kind of a title that I could search for through my computer files. And then within that, there's going to be multiple sequences, and each of those sequences needs a label. So this is, I know for a fact, this is the primer we use. So it's good that we have our primer labeled because that's very important for Singer data and interpreting where the position should be starting and where it should be ending. The primer is very important for that. I'm assuming one of these is a label of the sample, right? Yeah, okay, don't tell me what the sample is. Let's actually figure it out. And just like pet peeves, I always do 0.5. I always write things in the way that I have to write grants. So I always do a 0.5 margins, Arial font. Oh, let's change it. Arial font size 11, because this is how I write grants. The other font you might want to use for Sanger data or for alignment data is, and this is actually really important, Courier new. This is how I'll save my Sanger data because every letter in Courier new is the exact same thickness. So if you ever do an alignment, you'll see that the bases always match up regardless of the character if you use Courier new. But if you use Arial or Times New Roman, the A's, T's, G's, and the C's all have different widths. So you won't get a straight up alignment. So that's the other thing. Fonts are very important for like this kind of thing. Okay, so now we want to analyze like what it is. We want to figure out like what it is. So the context of this experiment, I'll just quickly describe this experiment. We had an unknown bacteria. We want to know what it is. And so what we did is we did 16S ribosomal DNA sequencing of an amplicon. And then now this is the Sanger data that reflects that. So this is a classical experiment of you have some unknown organism. You want to figure it out what it is. You just do some 16S PCR amplification, Sanger sequence it. Now what we do is when we have the text file, we know that the chromatogram is good. So we know that the sequence is good. Let's blast it. So let me pull up the blast. Have you ever done blast before? Yeah. Okay. So we're going to do nucleotide blast. So this is a local alignment algorithm. Let's just paste the sequence. And this is going to tell us essentially what it is, hopefully. So I'm not going to put in an organism because I don't know what it is. And I also want to check it against everything that's in the database. So if I leave this blank, organism optional, if I leave that blank, it will blast against everything. So I'm going to hit blast. Hopefully this is quick. Hopefully we're not just sitting here like do. And typically now, once the blast finishes, what we're going to look to do is we're going to look to highlight, color code certain features of this in my Word document. And just as a quick refresher, so you should have, this is now your data, your important data. And I can't be responsible for all my students' data. They need to be responsible for their data. So somewhere, again, to reiterate, and for everybody watching this, like you should also have this, you should have some folder that is like your initials data. And within that folder, you should have a subfolder where you have Sanger data. So maybe you have one subfolder that's agarose gels. Maybe you have another subfolder that's SDS page gels, which are proteins. And you have some other subfolders discriminated by different data types. One of those folders is going to be your Sanger data. And then within this, you're going to have subfolders of either date or experiment that you're working on. And then within this is which you would save these particular important data files. Okay, so that's just proper organization of these data. Okay, so we're getting some hits. So it's not getting 100% hit, which could he say either two things? It could say that sometimes if you're Sanger sequencing from a PCR amplicon, the PCR could have mutated it. All polymerases have a mutation rate, and it's highly likely that whenever you do a PCR, you're often sometimes mutating the template. That's very common. That could be one reason why we're not getting 100% hits, or it could be something completely unique that nobody has found yet. It's probably not something totally unique. Like, we've sequenced so much stuff at this point that's in the database that it's probably highly closely related to whatever this is. So essentially what you're seeing here is the percent like identity. So it's 98.13% identical in DNA sequence to Bacillus valensis strain N308 16S. And I'm just going to look through other things here. So I'm looking at essentially like, what are all the genera, the genus of the bacteria that it's most closely related to? And it's obviously hitting like most closest to Bacillus. So that's like uniformly across. So we know for a fact that this unknown, this is clearly Bacillus, like genera in terms of the genus. That's probably a fact. Oh, did not want to open that. That's a fact. Now, what species, we're not sure, right? Because the difference between 96 and 7% could be a single nucleotide polymorphism that could be a mutation. So do we know it's Bacillus valensis or subtilis? We're probably not quite sure. So if you force me to go down to species level based on this at this point, I probably would be like, I'm not quite sure. I'd have to sequence a couple more genes to be quite sure about the species from this data alone. Okay. But this is already like a big deal. Like, we know the genus now. That's what we wanted to figure out. So let's figure out now of this, what are the matches and what are the mismatches? So if we want to look at that, we just click the alignment. So here it's visualizing with the alignment of the Sanger data that we pulled with Bacillus valensis. Okay, so I'm just going to take this. I'm just going to pull this in here as the closest match. Just paste it in. And now if I ever want to find like this, I can control find it. There it is in my sequence. And if I wanted to, I close the chromas, but if I wanted to check on one of these to make sure like, oh, it actually had that deletion, I could copy, paste this and control find it in the chromas to check the chromatogram. Okay, I think this is a very general now overview, short video that describes enough about how to walk through and look through Sanger data. So I'm going to stop this. So after some private conversation, there is some data that I would like to show. We did get a hit that when we blasted, we got a result of no significant similarity found. So it wasn't matching anything in the database. So my next reaction was, well, if the text is not matching the database, let's now look at the chromatogram. So this is a real life example of when you would want to look at the chromatogram. And when we look at the chromatogram, you can see exactly what I was talking about, where in individual bases, you have a bunch of these N's. And N is where it can't determine what this base is because there's a high blue peak and there's a high red peak. And here there's a high green peak, a high blue peak, and a high red peak. So it has no idea of what the sequence is. And so this is essentially like bad sequence data. This is trash. So we got no results for essentially like the second hit. So I just wanted to show this as an example of bad sequencing data.